Welcome to another episode of Tom Kennedy Science, where we explore the universe. And today, we are going to explore the nucleic acids, both RNA and DNA. Yeah, before I get started, I like fantasy, including Game of Thrones. And, biologists, we see the world differently sometimes, right? So here's Daenerys with their hair, and you can see the single-stranded and the double-stranded RNA and DNA. Yeah, biologists, we often see the world differently than other people, but that's okay. So our lecture topics today are going to be what are the nucleic acids and their functions. And we're going to come back to that function part of it a couple of times there. We're also going to start with the structure of nucleotides, and then we're going to look at the structure of the actual nucleic acids. That's the polymers of DNA and RNA. And then we're going to spend some time on the differences between RNA and DNA. And this is where we're going to have a nice discussion on the importance of form and function in biology. Okay, so what are nucleic acids? Okay, these are one of the four macromolecules. We have the carbohydrates, the lipids, the proteins, and now we've got our DNA and RNA, which are the nucleic acids. These are polymers, and these large polymers, they're put together by condensation reactions, and they are found in the nucleus of our cells. And, well, they're used to store information, especially the DNA. And then we also have the RNA, which has many different functions, like a lot. But a lot of it is about gene expression and how you regulate gene expression. So it's a fascinating molecule. And these nucleic acids, you know what? They're everywhere. They're in all living things. So all life uses DNA, for example, to store genetic information. And the language of life, doesn't matter which of these animals you are, the language of life is nearly the same for everything on Earth. So we have two different types of nucleic acids. We have DNA, and we all know this. DNA stores genetic information, and that information can control the activity of cells, help it respond to the environment, and of course, very important for growth and reproduction as well. And all of that information is stored in the sequence of the building blocks, which are the nucleic acids. Okay, now the other one is RNA. RNA is a really versatile molecule. I mean, it carries out the directions of DNA. It helps make the proteins, but it also helps regulate how much of the protein, when do we make the protein, how much are we gonna make, right? So there's lots of stuff that RNA does inside of our cells. Like I said, it is a fascinating molecule with all of its functionality. And unlike DNA, it also comes in different forms. So it's kind of similar to some proteins. Proteins can be folded into many different shapes, and RNA can do that as well, which we will talk about. So you can have the messenger RNAs, the transfer or the tRNAs, and the ribosomal or rRNAs. These are all doing different things, and they have different shapes. Like I just said it, like the proteins, right? That fold them in different shapes, they do different things. And like I said, it's fascinating as to why they can do that. Okay, so this is really showing this is just a subset of what RNA can do. I'm not going to go over these because we don't need to memorize these in my class here today, but DNA stores genetic information. So as you can see, RNA, just what an awesome molecule. Okay, let's dive into the structure of nucleotides. So nucleotides are the monomers, okay? These are the building blocks of our RNA and our DNA, and they're basically made of three different parts. You've got a phosphate group. You can see that blue circle, that's a phosphorus surrounded by oxygens. Okay, that's our phosphate group, very hydrophilic. Then we have a 5-carbon sugar, a pentose sugar. And you're going, pentose? I don't know pentose. Think ribose and deoxyribose, but it's a pentose sugar. Pento means 5. And then we have one of the nitrogenous bases. And DNA and RNA both have 4 nitrogenous bases, but there's a difference between them as well. Okay, and there's our nitrogenous base. You can see the nitrogen in there. Okay, so these nucleotides, the carbons in both the nitrogenous base and the carbon are all numbered. If they're in the nitrogenous base, they're numbered like 1, 2, 3, 4, and 5. However, if you're in the sugar, we number them with a prime to designate the difference between the carbons from the sugar and the carbons in the nitrogenous base. So 1' is right there attached to our nitrogenous base. Then we have a 3' as you go around 1, 2, 3. And then at the top of the molecule that attaches to the phosphate, that is our 5' carbon. And this is really important because it gives directionality to the nucleotide, okay? Now, there are 5 of these nitrogenous bases, and they are adenine, guanine, thymine, which is found in DNA, cytosine, and uracil, which is found in RNA. So there's a difference right there. And we also have what is called activated nucleotides, and activated nucleotides have phosphate groups attached to them. And what that does is it allows these molecules to store a lot of potential energy. And whenever you make DNA during DNA replication or DNA polymerization, you're going to use the activated nucleotides, and you're going to do the same as you copy the information from DNA to RNA in a process called transcription. You're going to use activated nucleotides. And they're also important for chemical signalings. For any of you that go on into cell and molecular biology, you're thinking G-coupled protein receptors, because it's using a G protein, which is an activated protein for signaling. But one of the most important things of these activated nucleotides also is ATP, which is the triphosphate for adenine, or adenosine triphosphate. And that is incredibly important because it is the energy currency of all life. So much of the energy transfers that occur within our cells is done using ATP. Okay, so now, whenever we start to make RNA and DNA, these are the nucleic acids. We take our nucleotides, the building blocks, link them together in a condensation reaction, and it actually forms something called a phosphodiester bond. Okay, that's a lot, but let's take a look at it. So if you start with a carbon and you go way up here to our pentose sugar, you'll notice it goes carbon, then it goes an oxygen and a phosphorus. That's an ester, but it's got a phosphorus instead of another carbon in there. So that's a phosphoester. And then it goes carbon, oxygen, phosphorus, oxygen, carbon, COPOC. So it's a 2 ester bonds, hence diester. And because there's a phosphorus in there rather than a carbon, it is a phosphodiester bond. Okay, now that forms what is called the sugar phosphate backbone of our nucleotides. Sugar, phosphate, alternating back and forth between sugar and phosphate, and it is hydrophilic, and it can totally dissolve in water. And that's very important in terms of how this molecule interacts inside of our cells. Because we number those carbons in our sugar 1' to 5', okay, this gives directionality to the entire DNA or RNA molecule in a 5' to 3' orientation. So up there at the top, you would have the 5'. So our 5' carbon would be right there. You can see my little cursor. That's our 5' carbon. Our 3' carbon is there. So we have a 5' to 3' orientation for our DNA and our RNA. Okay, now let's take a closer look. That's the general form of a nucleic acid. Let's look at DNA. DNA is well known for having a double helix. Imagine taking a ladder and twisting it, and that forms a double helix where we've got, let's see here, we've got the sugar phosphate backbone right there, and then inside of that are the nitrogenous bases. And those nitrogenous bases, they pair up together. That is called base pairing. And I already talked about sugar phosphate backbone. And this anti-parallel, so they're going in opposite directions. Okay, so there's our DNA right there. That's our iconic double helical structure. The 2 strands are held together by hydrogen bonding in the middle between those nitrogenous bases. Okay, now here's something that's very important. There are 2 major classifications of our nitrogenous bases. Purines, which are the double ring structure right here, and the pyrimidines, which are the single ring structure. And I always remember pure as gold. Purines is adenine and guanine. Okay, so the purine is always going to pair with a pyrimidine, and A is always going to form 2 hydrogen bonds with thymine, and guanine is always going to form 3 hydrogen bonds with cytosine, okay? And we also call this Chargaff's rule that the number of purines in any DNA equals the number of pyrimidines. Okay, I've already mentioned this several times. Here's our sugar phosphate backbone, because there's a phosphate, there's our sugar, hence sugar phosphate backbone. And this is held together by covalent bonds. So where the 2 strands are held together by hydrogen bonds, the sugar phosphate backbone is held together by the much stronger covalent bonds that actually allows you to pull the 2 strands apart much more easily. And like I said, we have the phosphodiester bonds on there, and this is hydrophilic. So the sugar phosphate backbone interacts very nicely with water and actually turns those nitrogenous bases in toward each other. It also has an anti-parallel direction orientation. So imagine a divided highway. One side goes west, the other side goes east. Here we've got our 5' up here going down to 3'. And on the other side is the exact opposite. There's our 5' on the bottom going up to 3'. And those strands are running in opposite direction. And this is very, very important for DNA replication and transcription, which is something we will get into at a later time. Okay, let's take a look at RNA. RNA is single-stranded, unlike DNA, and it has a sugar phosphate backbone as well. And a difference is, whereas DNA, it can get into a couple different shapes, but most of the time it's in that very stable double helix, whereas RNA can fold into many different shapes, like this transfer RNA on the top, and as a result, it serves many functions. All right, here it is. Our theme in biology, theme across everything, right? Form and function, shape matters. The way that those differences between DNA and RNA really matter for how it functions and why they're different. Okay, so let's take a look. The differences between DNA and RNA. You may have memorized these differences in classes before. I know I diligently remembered them and memorized them, recited them on a test, had no idea why it was important. And we're gonna hit that hard here in a few minutes. Okay, so DNA has a deoxyribose sugar. So DNA stands for deoxyribonucleic acid. It uses the nitrogenous base thymine, and DNA is double-stranded. And when we look at RNA, it has a ribose sugar. It uses the nitrogenous base uracil, and it's single-stranded. Okay, what does that mean? Why does this matter? Let's take a closer look here. Okay, both RNA and DNA use a 5-carbon sugar. However, there's a difference between deoxyribose versus a ribose, and I'm actually showing a deoxyribose on that 2' carbon. And deoxy means without oxygen. So a deoxyribose sugar, if you notice on the one on the right, it says deoxyribose, it's missing an oxygen, okay? This makes that molecule more chemically stable for 2 reasons. It's less likely to be broken down by hydrolysis, and it's also less likely to be broken down by enzymes inside the cell called nucleases. There's a ASE, which tells you it's an enzyme. Nucleases break down nucleic acids, and this molecule with the deoxyribose is less likely to get broken down by that. So this molecule is more stable, and it also has structural stability as well. See, that lack of the OH, that hydroxyl group, it basically allows DNA to form the stable double helix structure. And it also reduces its flexibility. So that means that a long DNA molecule isn't going to move around a whole lot. It's going to assume that stable double-stranded double helix. Now, let's look at ribose sugar. Right there, you can see it's got a hydroxyl group, okay? It's less chemically stable. This molecule is easier to break down by hydrolysis, and those nucleases are more likely to break this down as well. Okay, so it also has more structural flexibility. So that OH there actually allows this molecule to fold into different shapes. Lots of different shapes, lots of different functions, right? Now, let's look at our nitrogenous base. On the left, we've got DNA, and on the right, we've got uracil used in RNA. And you'll notice that little up there at the top left of those, you'll see an H3C or CH3, which is a methyl group, okay? Thymine with that methyl group is actually more chemically stable than the uracil. There it is. There's our little methyl group right there versus not having it. Okay, DNA, that means, has a lower risk for mutations. Because here's another thing. There's one more of these pyrimidines, and that is cytosine. And cytosine can chemically, it can switch to uracil pretty easily. And for those of you interested in chemistry, it deaminates. It pops off a nitrogen, and the cytosine becomes uracil. Well, that's a real problem because we have all of these repair mechanisms that replace the wrong nucleotide. However, if you are changing cytosine to uracil, those DNA repair mechanisms won't detect that, right? So you want to use thymine, which is going to be much more stable than using uracil. All right. Now, here's the next one. Single versus double-stranded. DNA, as you know, double helix. It is very stable. It holds information. RNA is single-stranded. It folds into different shapes. Okay, here's our form and function. DNA stores information. This molecule is built for stability and longevity. Okay, this is important. The deoxyribose sugar, it is chemically stable. It is structurally stable. It allows the two strands to twist into this double helical structure. And by twisting the double helical structure into that a lot of double helical structures. By twisting it into the double helix, those nitrogenous bases move into the interior, and they're also protected from additional structural damage. Okay, and then we use thymine. It's more stable than uracil, which further reduces the risk of mutations. Now we get into RNA. Okay, RNA. This is a much shorter-lived molecule, and it's versatile with many functions. Our cells are constantly breaking down RNAs inside of our cells and repurposing those nucleotides over and over and over again. So this is less chemically stable. It's okay. They don't last as long inside of our cells. It is more flexible. By being more flexible, it can assume many different shapes. Hence, it's lots of functionality inside the cell. And uracil is cheaper to make than thymine, which is fine because we're going to use this over and over. If cytosine happens to switch into uracil, doesn't matter. You're just making a protein. If the protein doesn't work, take it apart again and rebuild it. You know, it's not going to like permanently introduce change into your genetic code. And there it is, because it's single-stranded like proteins, right? String of polypeptides of the amino acids. This is a string of nucleotides, has many different shapes. And I mean, check this out. You can have ribosomal RNA, tRNAs, and then there's our list of RNAs all over again. As you can see, this is just a partial list, but RNA is a very interesting molecule. There's been multiple Nobel Prizes have been given out for these RNAs and understanding what they do. All right, well, I hope you learned something about nucleic acids today. Until next time, stay curious. This is Tom Kennedy Science, signing off.